So, yesterday we went over transcription, and I kind of mentioned this, but I wanted to show you a little bit more detail. Wherever the code sequence is initiated is called the promoter. Wherever the sequence code ends, it's called the terminator. And there's always going to be a promoter site and or point and a terminator point. And that is the extent of the code interpretation. So there's going to be a promoter and a terminator. Today we're going to go over step 2. Step 2 of what? Raise your hand if you could tell me that. Step 2 of protein production. During step 1, or what is step 1 called? Who could remind me? This is a review. Transcription. It's where a message is transcribed. Who transcribes the message? Who transcribes the message? Raise your hand. What kind? mRNA, messenger RNA. Who creates the message? DNA, because DNA makes RNA. And when RNA interprets the message, where does it go?
Outside the cell.
Outside the cell. As soon as you leave the nucleus, outside the nucleus, we should say, the code begins to be interpreted. And as soon as you leave the nucleus, where are you headed? Raise your hand if you could tell me that. You're going to look for the ribosome. But where are you as soon as you leave the nucleus? Cytoplasm. So mRNA is traveling through the cytoplasm, and when it finds a ribosome, the ribosome is going to hold it in place. And that whole entire time, a code is being read. This code is called a codon because it is read 3 letters at a time. So let's look at the picture. If I am referencing RNA leaving the nucleus, I'm right here. Let me make my pen a little bit thicker. Right here. Okay? Where the code is initiating is right here. If that code is initiated at AUG, okay, that means the nucleotides are A, U, and G for that mRNA strand. tRNA is going to come in. It's going to be attached to an amino acid. Now, even though it says there that the tRNA is attached to an anticodon, that's what we call the nitrogenous bases on tRNA, anticodons. The 3 codes on RNA are codons. The 3 codes on tRNA are anticodons. So if my codon is AUG, who could tell me what the anticodon is going to be?
GUA.
AUG. What's my anticodon going to be? Raise your hand. UAC. The anticodon is going to hold the corresponding nitrogenous bases because that's who is attracted to AUG. AUG. Remember that they're always attracted to each other. Those molecules are attracted to each other. But that molecule is holding an amino acid. And what do amino acids do? Form protein. So the code is on mRNA. It says, bring me a specific protein. And the person or the object that brings it is tRNA, which has the corresponding nitrogenous bases. I'm going to show you an animation, and hopefully that'll make a little bit more. So here's an animation. If we go back to what I was explaining before the animation, which you could access by clicking on the links that I'm going to post on files on Canvas. Here's mRNA. There's the start right here where I'm going to point to. Okay? And what's this? If I circle this, who could tell me what I'm circling? Raise your hand.
The thing that has,
I can't, no more things. tRNA. That thing is tRNA. What's attached to tRNA? Amino acid. This is the amino acid, this weird structure here. Okay? What's at the bottom of tRNA? Nitrogenous bases. What do we call those 3 nitrogenous bases at the bottom of tRNA? Anticodons. Okay? Now, so there's 64 codes, yet there's only 20 amino acids, and you do not have to memorize which amino acid goes with which code. This chart will be given to you on the test. What I do want you to know is, where do the 64 codes come from? Raise your hand if you could give me that answer. So you have 4 nitrogenous bases, and each code is 3 letters. So 4 to the 3rd power, there's a possible 64 types of codes. UUU calls for phenylene. So does UUC. Since there's only 20 amino acids, codons, 2 different codons or 3 different codons could call 1 amino acid. Okay? So, like GGU, GGC, I'm pointing right here, all call for the same amino acid. In this case, there's 4 codons which call for 1 type of amino acid. And you don't have to know that. I will put this chart on the test.
What about the ones that say stop?
The ones that say stop means stop making protein.